Hi, I'm Kristen Sharpe. I'm a master's graduate student at the Virginia Institute of Marine Science, and my Virginia Sea activity deals with zooplankton, their fecal pellets, and it teaches students why zooplankton and their fecal pellets are important in the global carbon cycle. So what students are doing is they're helping me solve the mystery of where all of my samples came from. So students are told that I went to 10 locations around the world and set sediment traps, which are essentially funnels that collect all the drifting material that drifts into the mouth of the funnel and concentrate them into bottles. And so things like discarded fecal pellets can drift down into these sediment traps and we can collect them and study them. And so students are working in small groups. They're given a bag containing fecal pellets that can be either candy or bean, pipe cleaner, and marshmallow focused. So I have two options for that as far as cost and reusability. So the students are given a bag of fecal pellets. And what they do is they pour them out onto their desk. For example, this one contains what is supposed to represent Antarctica in the summer. So there's a variety of pipe cleaners that represent Antarctic krill pellets, as well as marshmallows, which represent salp fecal pellets. And so what they do is they dump out their contents. They use the map that's included to make a hypothesis about where they think that their sample bag came from. Then the students are tasked with sorting out the different fecal pellet shapes and sizes. They count how many of each type they have. They also weigh them. So they weigh them on pre-weighed coffee filters on either a triple beam balance or a digital scale or whatever you prefer as far as measuring weights goes. They record this data on a data sheet that they turn in as a group. There's a variety of questions to guide them through the thinking process and to really drive home the structure of the scientific method. And so then the students are using a dichotomous key to identify the source species for each of the zooplankton fecal pellets that are in their bag. And so after they're all done, they compile all of their data, they come together as a group, talk about where they think each of the bags came from. They have a discussion about it before the teacher eventually reveals the correct answers for where each bag came from. And then the students work together as a class to graph the total weight of fecal pellets in each of the 10 locations to show different rates of fecal pellet production and carbon cycling in a variety of locations around the world. And then in addition, I've provided some other optional activities that expand on this, including graphing community composition, so pie charts that show the relative amount of each type of zooplankton in each area, as well as including real fecal pellet photographs from my research that students can use an included scale to measure the size of the fecal pellets and get a feel for what my research is actually like. Thank you.